Genetic inheritance and genetic control. We are going to discuss question in this chapter. Question number 1. An allele is, the answer would be an alternate form of a gene. Basically, when you have, for example, a homologous chromosome, okay, a chromosome to a set of chromosome where you inherit from your father and also from your mother, and then for example here you have a gene, okay, a gene, this region here along the chromosome codes for your eye color, okay, for your eye color. So the gene will lead to the expression of your eye color. So eye color will have many eye color, okay, so there is blue eye color, there is brown eye color, there's green, alright. So all this variation is determined by the different allele that are present. Same goes to gene that codes for blood type, okay. So genes that can code for blood type, for example, you have blood type A, blood type B, okay, blood type AB, blood type O. So all this variation of blood type are determined by the different alleles for the character of blood type. So there's allele IA, IB, I or IO. So this allele will lead to the expression of different traits, okay. So question number 2, location of a specific gene along a chromosome is called a locus. So like what I've drawn here, okay, the specific position along a chromosome where certain genes are located, we call it as locus. So for example, here you have the locus for the gene that code for eye color. Here you have a locus, the location of the gene that code for or that expresses blood type, okay. Question number 3. To determine whether the genotype of a purple seed plant is homozygous dominant or heterozygous, Irfan should do a test cross between the plant and the other one is homozygous recessive plant. Okay, so the answer will be A. So you have a plant, you have a purple plant with, you have a plant that has purple flower. So purple is a dominant character. So when purple is a dominant character, the individual can either be homozygous dominant, the allele will be capital P, or the individual can also be heterozygous, okay, capital P small p. Alright, so you don't know whether the plant is either homozygous dominant or heterozygous. So therefore, you have to cross the plant with the unknown genotype with an individual that is homozygous recessive. Homozygous recessive, the phenotype of an organism that is homozygous recessive, you know exactly, or you can observe the phenotype. For example, pea plant that has white flower, okay, pea plant that has white flower, you for sure know that the plant with white flower is homozygous recessive because it only has one genotype, which is small p. So in order to determine the genotype of the unknown plant, you have to cross the plant with homozygous recessive. So for example, if the plant is homozygous dominant, when you cross with a homozygous recessive individual, the plant that has white flower, okay, so the F1 generation or the offspring would produce 100%, okay, 100% purple flower. Okay, so this is the gamete for the first parent. This is the gamete for the second parent. Okay, so when the gamete fertilizes each other, the F1 offspring will be heterozygous, which is 100% purple. If the individual is heterozygous, so when you cross the unknown individual that you think that it is heterozygous, okay, so you cross with the homozygous recessive plant, the plant that has white flower. So you draw the gamete, okay, this is the gamete with the recessive allele, and then for this individual, for example, this is the male plant, this is the female plant. So this male plant will have two type of gamete, Okay, so one with the dominant P allele, the other one with the recessive P allele, and then you cross these two plants together. Alright, so this and this will produce heterozygous individual, and this will produce homozygous recessive. So it means that 50% of the offspring is purple, and another 50% of the offspring will have white flower. So it means that if the offspring produces recessive trait, so the unknown individual will be heterozygous. If all the offspring produces dominant trait, so the unknown individual is homozygous dominant. Okay. Next, question number 4. Albinism, which is lack of pigmentation in human, result from an autosomal recessive gene, which is A. Okay, the recessive A allele results in the phenotype being albino. Okay, so in order for a person to be albino, the person is homozygous recessive. Okay, homozygous recessive. Two parents with normal pigmentation have an albino child. Okay, what is the probability that their next child will be albino? So this when two people, okay, that has normal pigmentation on their skin, they have normal pigmentation, but when they have child, they find out that their child is albino. Okay, so it means that these two individuals are actually heterozygous because they carry the recessive A allele, that will exhibit the albino phenotype. Okay, so for example, these two individual, okay, that is they both are heterozygous. So this is the father and this is the mother. Okay, the parental generation, and then the gamete, okay, will produce gamete with the dominant A allele, gamete with the recessive A allele. So that's the mother. Okay, the gamete will be dominant A and another one with a recessive A allele. Okay, and then you do to make it easier to understand, you put it in a Punnett square. Okay, alright, so and then you just match all the gametes together to form the genotype of the offspring. So this will be normal. Okay, normal, homozygous dominant. Capital A, small a is heterozygous. These are normal also. They are heterozygous, they are carrier actually. This one also heterozygous, they are normal but they are carrier. And then the last one is homozygous recessive, and this is the albino child. Okay, so there are four possible outcome. Okay, you have the one homozygous dominant, two heterozygous, to one albino. Okay, so the chances is 1 out of 4. Okay, 1 out of 4. Okay, so the answer will be A. Question 5. The term test cross refers to, the answer will be C, which is breeding an individual of unknown genotype with a homozygous recessive. This individual with unknown genotype, the individual will exhibit dominant phenotype. Okay, dominant phenotype, but you do not know whether the individual is either homozygous dominant or heterozygous, but the individual will exhibit dominant phenotype such as purple flower, for example. So you have to cross with another individual that exhibit recessive trait or recessive phenotype, because for sure you know that the genotype of that individual will be homozygous recessive. In this case, for example, for flower color is white. Okay, that is a test cross. You cross with an individual that has homozygous recessive. Okay, that is homozygous recessive that portrays a recessive trait. Question 6. In tomato plants, the allele for purple stem P is dominant to the allele green stem. Okay, so you extract out the information. So P for purple. Purple, so it means that the genotype for purple can either be homozygous dominant or heterozygous. Okay, so this is one allele, and then you have another allele which is recessive allele, which will express green stem. So in order to produce green stem, the individual has to be homozygous recessive. Okay, so this is for one gene. And another gene that has two allele, which is allele that determines whether the stem will be hairy or smooth. Okay, so this is the first gene. So you have the second gene that is controlled by the H allele. So you have the dominant H allele for hairy stem. So the genotype can either be homozygous dominant or heterozygous. Okay, and then recessive allele for smooth stem will be homozygous recessive. Okay, so the question, the following cross was carried out. Okay, which is the cross between an individual with this genotype and another individual with this genotype. So 32 offspring were produced from this cross. How many of these offspring will be expected to have purple smooth? Okay, purple smooth. So smooth must be homozygous recessive. Purple can either be homozygous dominant or heterozygous. So you have to do a cross. Okay, between these two individuals. So for the let's do for the second parent first. So the gamete for the second parent will be small p small h. Okay, but the gamete for the first parent here, it is a bit complicated, where it produces 4 different gamete, which is capital P, capital H, okay, capital P, small h, and then the other one you have small p, capital H, small p, small h. Okay, so you do a crossing between the two parents. Okay, so to make it easier, you may construct a Punnett square. Okay, where on this is the gamete here for the, let's say this is the female parents, this is the male parents. So on this side female, this side is male. So male will be PH, the gamete PH, capital P, capital P, small p, capital H, and then the last one small p, small h. Okay, and then you just combine the gametes together. Okay, which will produce capital P, small p, capital H, small h, capital P, small p, small h, small h, small p, capital H, small h, small p, small h, small h. So the question wants for purple and smooth. So which of this genotype will be purple and smooth? Okay, so this will be purple and hairy, purple and smooth. So this one here, this is purple smooth. Okay, this one will be green hairy, green smooth. So you want this one only. Okay, this. So it is the probability will be 1 out of 4 possible phenotype produced in the offspring. So 1/4 you will have to times by 32. So the answer will be 8. Okay, so the answer will be B. Okay. Question 7. A cross between homozygous purple flower. Okay, homozygous purple flower, capital P, and homozygous white flower. Okay, white flowered pea plant. Okay, which is small p, because it says homozygous. Okay, so results in offspring with purple flowers, obviously lah. Okay, so the offspring, the F1 generation, this is the parental generation, so produces heterozygous individuals. So 100% of the F1 generation will be purple. So because the F1 is heterozygous with the presence of dominant phenotype, and it expresses the offspring expresses dominant phenotype, so it means that the inheritance is complete dominance due to the presence of the dominant P allele in the F1 generation, the genotype of F1 generation. Okay, complete dominance. Okay, choose question 8. Choose the correct pair from the following table. So epistasis, example is ABO blood group. So this one is obviously wrong. So for ABO blood group, it involves multiple allele and also codominance. Okay, number 2, codominance, roan fur in cattle. So this one is the correct answer, where the fur color in cattle will exhibit roan color, where you have the cow having both red and white. Okay, white patches in the offspring. Okay, so it means that both of the allele, the capital or the dominant R allele and also the recessive R allele will be expressed in the offspring, produces cow with patches of red and white fur, which is roan. Okay, this is codominance. Both allele will be expressed. Okay, so multiple allele, example snapdragon plant for flower color. So snapdragon plant for flower color, actually it exhibit incomplete dominance, where you have red flower crosses with white flower, which produces offspring with intermediate phenotype, which is pink. Okay, so intermediate phenotype is incomplete dominance actually. So this one is not multiple allele but incomplete dominance. The last one is skin pigmentation. So skin pigmentation is not incomplete but it is polygenic inheritance, where one character, which is skin color, is controlled by 2 or more gene. Okay, 2 or more genes. Question number 9. When the phenotype of both allele are exhibited in the heterozygous, it refers to codominance. For example, a person of blood type AB, so the genotype is IA, IB. Okay, so both of these allele will be expressed in the individual. So for example, the red blood person, the red blood cell of that person will have both the A antigen and also the B antigen on the surface of red blood cell. So codominance, co means together. Both of the allele will express together in the offspring. Because both allele will be expressed fully. Okay, expressed fully. Okay. So that is codominance. Both allele will be expressed fully in the offspring. Alright. Number 10, a cross between two pure breeding lines. Okay, pure breeding lines mean that the parents is in the condition of homozygous. It can be homozygous dominant or homozygous recessive. Okay, so a cross between two pure breeding lines, one with red flower, okay, and the other one with white flowers, produces F1 offspring that are pink. Okay, when F1 progeny are self-fertilized, a 1:2 to 1 ratio of red to pink to white flowers is produced. Determine the genetic inheritance consistent. Determine the genetic inheritance consistent with these results, which is the answer would be incomplete dominance. Okay. So the parental generation they are pure breed. This is the male parent, this is the female parent. For example, you have the other one is the male parent. Okay, so let's say the female parent is red, the male parent is white. Okay, so the F1, the gamete will be capital R, and then the other gamete will be small r. So the F1 generation will be heterozygous. Okay, heterozygous, having both dominant and recessive allele, and produces a phenotype which is pink. Pink, okay, pink is the phenotype that is intermediate of both parent. So when the offspring produces phenotype that is intermediate, it is incomplete dominance. So the ratio, okay, so when you allow the F1 generation to self-fertilize, okay, so you do you do another heterozygous individual that is pink, and then the outcome will be, okay, capital R, small r. Okay, this is the male, for example, this is the female parent, capital R, small r. So capital R would produce red, capital R, small r would produce white, pink. Okay, pink, capital R, small r, pink as well. And then the other one is small r, which is white. So the genotypic ratio would be what? 1 capital R homozygous dominant to 2 heterozygous to 1 homozygous recessive, which is the same as the phenotypic ratio. Phenotypic ratio would produce 1 red to 2 pink, okay, to 1 white, right? So that is the phenotypic ratio, this one here, 1:2 to 1, which is the ratio for genotypic ratio and also the ratio for phenotypic ratio. So it is incomplete dominance. If you observe for question number 11, it is similar to question number 10, and the answer should be incomplete dominance as well. Okay. Question number 12. Which blood type would not be possible in children with AB blood type mother and A blood type father? So the mother is blood type AB. So the genotype will be IA, IB. The father, okay, which is blood type A. So the father can have either the genotype IA, IA or IA, I. Okay, so the offspring can only be, okay, the offspring can only be can only have blood type A, this one, this allele with this allele, or this allele with this allele. Okay, so or the offspring can be blood type B, this allele with this allele. Okay, so the offspring can also be blood type AB, this allele with this allele, IB allele with IA allele. Okay, so and then that's it. The offspring cannot be blood type O because the mother does not have any recessive I allele. Okay, so the answer will be O. Okay, so number 13. Which of the following example of human inheritance can be explained by polygenic inheritance? Polygenic inheritance, where poly means many genes. Okay, one character is controlled by 2 or more genes. Okay, 2 or more genes. So for example, is skin pigmentation. So another character that is controlled by polygenic inheritance, such as for example height or weight. So these are the character that you can count, and then you can plot the data to form a graph, and the graph will give a normal distribution. Okay, so for example, skin pigmentation, there are people that are very dark on this side of the graph, there are people that have fair skin tone, and on average there's quite a large number of people that has intermediate skin tone, for example. Okay, same goes to height and also weight. Okay, number 14, what is the difference between DNA replication in prokaryotes and also eukaryotes? So the answer will be, if we look at one by one, replication is conservative in prokaryote. There are no Okazaki fragment in prokaryote. There is only one origin of replication in prokaryote. So the answer will be C. Okay, so prokaryote, because the amount of DNA is not as much as eukaryote, because prokaryote is a simple organism. Okay, so for prokaryote, they only have one origin of replication, but as for eukaryote, they have many origin of replication, because this is to speed up the rate of DNA replication, because the amount of DNA is much more in eukaryotic cell. Figure 1 shows a pedigree of the family that have cystic fibrosis. So this is actually a pedigree that shows the inheritance of cystic fibrosis. So it shows the inheritance of the disease over 3 generations. The first generation are the grandparents. The second generation are the parents. Okay, the parents and the uncle and aunts. And the third generation are the grandchildren. Okay, the 3 siblings. All right, so if you observe, the grandmother has the disease, okay, and the grandfather is normal. So the first couple here of the first generation produces children in the second generation that are all normal. So it means that they are normal, but they are carrier for the allele that causes this that causes the disease cystic fibrosis. For example, we give the allele that controls cystic fibrosis as small a. Okay, so this is actually the pedigree that shows the inheritance of recessive trait. Okay, because the first generation, the grandmother has the trait, but in the second generation the trait is missing, okay, and appears again in the third generation. Okay, so that means the second generation are the carrier for the recessive allele. So the answer would be, okay, choose the correct statement based on the information given in the question. Okay, in figure 1, so the answer would be A. All individuals, okay, all individuals in the second generation are carriers. Okay, carriers for the recessive allele. This individual, the male here that marries the first daughter of the couple, okay, is also heterozygous. So the genotype will be something like this. Okay, capital A, small a, and then the chances of having a child with the disease is 25%. Okay, 25%. Question 16. The function of DNA polymerase is to build a strand of DNA using DNA as template. So during DNA replication, where you have the DNA double helix will be unwound, okay, to form a replication bubble. Okay, to form a replication bubble. So imagine this is the origin of replication. So the start of replication produces a primer, okay, a primer, RNA primer using primase. So this produces a 5' end on the primer and a 3' end on the same primer. So and then will come the enzyme. The enzyme involved is actually DNA polymerase 3. Okay, the function of DNA polymerase 3 is to add DNA nucleotide, okay, nucleotide at the 3' end of the primer, okay, or the existing strand. Okay, so the addition of DNA nucleotide is based on the DNA sequence on the template. So these are the template DNA template, the parental DNA, okay, that becomes the template to synthesize the new DNA strand using DNA polymerase 3 to add DNA nucleotide to elongate the DNA strand during DNA replication. So number 17, which of the following bonds are between complementary strand of DNA? So you have two strands of DNA that runs in the opposite direction or anti-parallel. So on this strand you have the 5' end, on the end you have the 3' end, 3 with 5, 5 with 3. So the direction will run from 5 to 3, from 5 to 3, anti-parallel. Okay, so let's say on one strand you have adenine, the other will pair with thymine. On this strand you have guanine, the other will pair with cytosine. So the two DNA strands will be held together by hydrogen bonds. So the hydrogen bond between adenine and thymine will be 2 hydrogen bonds. The hydrogen bond between guanine and cytosine will be 3 hydrogen bonds. Okay, so the answer will be A. Hydrogen bonds between the nitrogenous bases. Okay, question 18. Replication of DNA will involve all these enzymes except, okay, the answer will be RNA polymerase. RNA polymerase is the enzyme that will be required during transcription to produce RNA based on DNA template. Okay, so all the other enzymes are required during RNA replication, which is primase, to produce primer, topoisomerase to reduce a strain ahead of the replication fork, and then DNA polymerase 3 is to add DNA nucleotide according to the sequence on the DNA template. Okay, so number 19. A strand of DNA with the sequence 5' AACTTG 3' will have a complementary strand with the following sequence. Okay, which is one? Which one? The answer will be D. Okay, so if you look at the sequence 5' AACTTG 3'. So this is the first strand. So the second complementary strand will read 3', 5 matches with 3, A with T, T, G, A, C, 5'. So this is the sequence on the second strand, which is this one here. So question 20. If a sample of DNA is composed of 10% adenine, okay, 10% adenine and 40% guanine, its composition of thymine and cytosine must be, okay, adenine, the amount of adenine must be equal to thymine because adenine has to pair with thymine. So thymine must be 10% as well. Okay, so guanine pairs with cytosine, so the amount of cytosine will be the same as guanine, which is also 40%. So the answer will be A. So question 21, a fragment of DNA contains 22% thymine. Okay, calculate the percentage of cytosine proper, okay, in the DNA. Okay, so the information given here is just enough for you to calculate the percentage of cytosine. Okay, so the total amount is 100%, the total amount of DNA, right? Okay, so thymine pairs with adenine, so the amount of adenine will also be 22%. So total together, adenine and thymine will be 44%. Okay, so the rest, okay, will you will have to minus 100 with 44. You will get how much? So you will get 56. So 56 is the amount for both guanine and cytosine. So guanine pairs with cytosine, so you have to divide the amount by 2. Okay, you have to divide the amount by 2 because the amount is equal for both guanine and cytosine. So 56 divided by 2 is 28. So the amount of guanine and cytosine will both be 28. Okay, so cytosine will be 28. So if we were to check the answer, okay, so 22 adenine plus with 22 adenine, 28 guanine and 28 guanine, sorry, cytosine, the total amount will equal to 100%. Okay, so question 22, the following events occurs in DNA replication. In which order do these events takes place? So you have to analyze these orders to make sure that they are in the correct sequence. Okay, so for DNA replication to occur, first DNA molecule uncoils and separate. So now this is number 1. Second, the second event would occur is after the parental DNA unwinds, okay, to be separated to become the template, then free DNA nucleotide align with complementary nucleotide on each strand, on each of the template strand. So this the red line here indicates free nucleotide being added, okay, being added to the strand. Okay, so this is number 2. And then bonds between complementary bases formed. Okay, so the bond between the complementary bases will be hydrogen bond. So this is number 3. And the last one is phosphodiester bond will form between the bases. So the answer will be number 1. Okay, number 1, number 4, number 3, and number 2. Okay, which is C. Okay. Question 23. Which of the following is not the characteristics of genetic code? Okay, the first one, a start codon is AUG, so this one is correct. In order to initiate the process of translation along the mRNA, okay, you have the RNA sequence, and it must start with the codon AUG, which code for the amino acid methionine. Okay, so this is the start codon to initiate the process of translation, the process of synthesizing the polypeptide chain. Okay, the process of translation. So the first statement is correct. Number 2, there are 3 codons out of 64 which do not encode for any amino acid. So these 3 codons are known as stop codon, and you have to remember the sequence of these 3 stop codon, which are UAA, UAG, and also UGA. So you have to remember this 3 stop codon. The 3 stop codon indicate the termination of a translation process, the termination of the translation process. Okay, so the third statement, so the second statement is correct. No, a non-overlapping codon is when each base in the sequence is read only once. For example, you have your long mRNA strand. Okay, so the mRNA strand will have a specific sequence. For example, it can read as AUG as the start codon, and then you have another 3, such as for example CCG, UG, CCUG. Okay, so these are the specific an example of a sequence found on mRNA. So every 3 bases, okay, we call it as codon. Okay, so each codon will be only read once to specify or code for a certain amino acid. Okay, so the codon will code for the certain amino acid in the process of synthesizing the polypeptide chain. Okay, so each codon can only be read once, none overlapping. So the statement C is correct. D, they are degenerate, meaning that some amino acid are specified by one codon. Okay, so the statement is incorrect because amino acid can be coded by some can be coded by one amino acid, some can be coded by 2 codon. Okay, some can be coded by 3 codons. Some amino acid can also be coded by 4 codon. So if you refer to your slide note, slide 11, it gives you a codon table of mRNA. For example, if you look at in your codon table, the amino acid valine, it is coded by 4 codon, which is GUU, GUC, GUA, GUG. Okay, so statement D is incorrect. So there are amino acid that is coded by 1, by 2, by 3, or by 4 codon. Okay, so number 24, which enzyme is involved in the transcription of DNA? Okay, so transcription of DNA involves the synthesis of, for example, mRNA or rRNA or tRNA. So the DNA will become the template. Okay, so this is the DNA. Okay, so the DNA will separate to become the template to synthesize the mRNA. So in order to synthesize the mRNA, it requires the enzyme RNA polymerase. Okay, so the RNA will be synthesized in the direction of 5 to 3. Okay, 5' to 3' direction. So it means that the DNA that becomes the template must run in the must be 3' and 5' over here. So they must complement the mRNA. The strand must complement with the DNA strand. Okay, so these other enzyme, ligase, helicase, and DNA polymerase are the enzyme involved during DNA replication. Number 25. For a DNA triplet code CAT, choose the correct corresponding mRNA and tRNA sequence. Okay, so this is the DNA template. It reads CAT. Okay, so the DNA becomes the template to synthesize mRNA. Okay, so mRNA will the sequence on the mRNA will read C is G, A with U, T with A. From mRNA, it will complement with the anticodon, okay, anticodon on tRNA. So the anticodon on tRNA will read as G matches with C, U with A with U. Okay, so the answer will be which one? mRNA is GUA, the tRNA will be CAU, so the answer will be C. Okay, so question 26, if DNA strand that reads, okay, TAG, GGC, ATG undergo transcription. So this is the DNA strand that becomes the template to synthesize the mRNA. So the sequence of the mRNA will be which one? So you just match. Okay, so this is the DNA. mRNA will read as T with A, U, C, G, U, A, C. So which one? A, U, C, G, U, A, C. So the answer will be D. Okay, the answer is D. Right? So question number 27. 3 nucleotide sequence on mRNA that code for specific amino acid is called obviously a codon. Okay, the codon on mRNA that reads AUG will bring specific amino acid, which is methionine, for example. Okay, number 28, a template of DNA that reads 3' TAG 5' undergo transcription to synthesize mRNA sequence, which is okay. So the template on the DNA template must read 3. Okay, 3 because in order to synthesize mRNA, the synthesis of mRNA must run in the 5 to 3' direction as well. Okay, same as the DNA replication. The synthesis of polynucleotide must occur in the 5 to 3' direction. Tak kisahlah dia mRNA ke ataupun DNA ke, the synthesis of polynucleotide must run in the 5 to 3' direction. So the template of DNA, okay, dia akan start dengan 3 dan 5. Okay, 3 dan 5. DNA, the strand is 3, on the other strand is 5. So that it can synthesize mRNA starting with 5' first. Okay, 5 and then 3. Okay, so the DNA, the strand will read TAG. So on mRNA will read what? T with A, U, C. So the answer will be A. Okay, so 28 is A. Okay, so the next question, what is the maximum number of codons that could be produced from 4 different bases? So the bases is ACG, the bases on mRNA basically. Okay, so how much codon can be generated from these 4 bases? So for each codon, how many bases? 3, right? Okay, so 4 to the power of 3 will equal to 64 different codon that can be possibly generated from the 4 bases. So 64 codons possible that will code for 20 different amino acid. Okay, so the answer will be C. Question 30. A terminator sequence in DNA is best described as, okay, terminator sequence on DNA. So you have a DNA molecule that serves as a template to synthesize mRNA. Okay, so the terminator sequence will terminate the process of transcription, will terminate the process of transcribing the mRNA. So the answer will be C. Okay, a sequence of bases at which RNA polymerase stop transcribing, stop producing the mRNA. Okay, so the enzyme that involved is RNA polymerase. So when RNA polymerase encounters a terminator sequence on DNA, so this indicate the process of translation. The process of transcription must stop. Okay, must be terminated. Question 31. The genetic information for protein synthesis. Genetic information is referring to the information in DNA which is in the nucleus. For protein synthesis is the process of translation. Okay. The genetic information for protein synthesis is carried to the ribosome. Ribosomes are in the cytoplasm. The process of translation also occurs in the cytoplasm. So the information in the DNA molecule, okay, that code for the synthesis of the specific protein. So this is, for example, gene 1, which code for the synthesis of enzyme 1. Enzyme is a protein, so the information is in the DNA. Okay, so this information in the DNA molecule is really big. The process of translation occurs in the cytoplasm. So therefore, the information in the DNA must be copied first to the process of transcription. Okay, so the information is copied into a smaller molecule. We call it as mRNA. To produce mRNA is through the process of transcription using DNA as template. So the mRNA now contains the information to synthesize the protein. The synthesis of protein occurs through the process of translation, which occurs in the cytoplasm. Okay, so that means the answer will be mRNA. The genetic information in the DNA for protein synthesis, which is translation, is carried to the ribosome through mRNA, because mRNA, the molecule is smaller, it is single strand, it can migrate from the nucleus into the cytoplasm to undergo translation to produce the protein. Number 32, no observable effects on the phenotype is the result of silent mutation. So there's basically 3 types of mutation that occurs if a sequence on codon is changed. Okay, so you have the silent mutation, missense mutation, and nonsense mutation. So for example, the codon on DNA, if the codon on DNA undergoes mutation, so the codon on mRNA will also change. Okay, when the codon changes, it can lead to these 3 different mutations: silent mutation, missense mutation, and nonsense mutation, if the sequence on the codon change. Okay, if the basis on the codon changes from the original codon. Okay, so for example, for silent mutation, you have an example of silent mutation such as okay, you have a codon which reads CUU. Okay, CUU. So CUU will code for the amino acid leucine. Okay, leucine. Sorry, leucine, L-E-U, leucine. Okay, so if the third codon changes, okay, if the third codon changes to, for example, a C, okay, instead of U it code for C, CUC, okay, it still code for the same amino acid. If the third codon also changes to A, which reads CUA, it still code for the same amino acid. This is what we call it as silent mutation. So the codon changes but still code for the same amino acid, so therefore it does not affects the expression of that certain trait. Okay, as for missense mutation, it leads to the codon being changed and code for a different amino acid. For example, here you have for missense, for example, you have the amino as the codon that reads CAA. Okay, CAA, which code for the amino acid GLN. Okay, I'm not sure the actual name for the amino acid. Okay, so CAA, if you change the sequence, the sequence into CAC. Okay, CAC. Instead of CAA, it changes to CAC. So this gives a different amino acid, which is his. Okay, histidine, if I'm not mistaken. His. Okay, so this is what we call it as missense mutation. The mutation occurs and code for a different or wrong amino acid. If nonsense mutation occurs, it changes to a stop codon and terminate the process of translation. Okay, it produces a much shorter polypeptide chain that causes the protein might not be functional. That is nonsense mutation. It changes to a stop codon. Okay, a stop codon. Frame shift mutation is a different story, where it involves deletion or insertion of base in between the codon. That leads to frame shift mutation. Okay. Question 33. Which of the following enzyme catalyzes the formation of peptide bond between two amino acids during translation? Okay, so the process of translation occurs in 3 stages, which is the first stage is initiation, the second stage is elongation, and the third stage is termination. Okay, termination. Okay, so the formation of peptide bond between two amino acids during translation occurs in the second stage of translation, which involves the enzyme peptidyl transferase. Okay, so first of all, you have to know the structure of a ribosome. So this is a ribosome. The ribosome consists of the large subunit and the small subunit. Okay, in between the subunit, you have the mRNA going through the two subunit. Okay, so this is the 5' end of the mRNA. This is the 3' end of the mRNA. Okay, so in the large ribosomal subunit, it has 3 sites. Okay, the A site, the P site, and also the E site. Okay, A, P, E. Okay, so in the P site, okay, is the place where the tRNA holding, okay, the polypeptide chain, the elongating polypeptide chain. So here you have the amino acid of the growing polypeptide. Okay, so A site is where the tRNA, the new tRNA comes and bring the next amino acid to be added to the growing polypeptide chain. Okay, so the function of peptidyl transferase is to form peptide bond between the last amino acid on the tRNA that sits in the P site, and then it transfer to the amino acid that sits in the A site. Okay, the transfer then involve the formation of a peptide bond. Okay, a peptide bond between the amino acid in the A site and the last amino acid in the P site. It is transferred from P to A and involve the formation of a peptide bond. Okay, so that is the elongation stage of translation, which involve peptidyl-transferase forming peptide bond between the amino acids in the polypeptide chain. Name the bond that joins arginine and alanine in figure 2. Arginine is this amino acid, alanine is this amino acid. So the two amino acids are basically joined by peptide bond. Okay. Question 35. Figure 3 shows a eukaryotic translation complex during elongation stage. Which of the following description of the diagram is not correct? So if you look at the statement A, which is this one, A is anticodon of a tRNA molecule. So this is the tRNA molecule that has the anticodon that matches the codon on mRNA. So this is mRNA molecule. Okay, mRNA. Okay, so the anticodon matches the codon. The anticodon on tRNA matches the codon on mRNA. So the statement A is correct. B, this one here. Okay, so B, location of mRNA cap structure. So the mRNA has two ends, which is the 5' end and also the 3' end here. Okay, so at the 5' here is you have the 5' cap. Okay, at the 3' end you have the poly A tail. The function of the 5' cap and also the poly A tail is actually it has several functions. The first one is to facilitate the mRNA moving into the cytoplasm. Second is to protect the mRNA from hydrolytic enzyme in the cytoplasm. And the third one is to help the mRNA attached to the ribosome. Okay, attached to the ribosome. And then so the statement is correct for B. And then if you look at statement C and D, it says here for statement C, the tRNA which exits from the E side of ribosome, so which is this one. Actually, this here, okay, is the A side. Okay, is the A side. The middle side here is the P side. Because it has the tRNA that has the long polypeptide chain, the growing polypeptide chain. The A side has the tRNA that brings the new amino acid to be added to the elongating polypeptide chain. On this side here you have the E side. E side is where the tRNA exits the ribosome. Okay, so basically for statement C it is incorrect. Okay, so because this is not the E side, this is the A side. Okay, so the tRNA which exit from the E side of ribosome. So statement C is incorrect. If you look at statement D, next amino acid to be added to the growing polypeptide chain, which is this one. Okay, D is the tRNA that has been used, that has transfer that has transferred amino acid to the growing polypeptide chain, and it exits the ribosome through the E side. So if you look at this question, actually both C and D statement is incorrect. Okay, so dia tertukar. Okay, so sepatutnya D ni adalah the tRNA which exit from the E side of ribosome. Yang C ni adalah the next amino acid to be added to the growing polypeptide chain. So both statement C and D is incorrect. Dia tertukar. Okay, so for this question it has two answers, C and D. Identify which of the following is correct about point mutation. Okay, about types of point mutation. So the first one, silent mutation is description code for a different amino acid. So this is wrong. Silent mutation occurs and still code for the same amino acid. Okay, it code for the same amino acid. So this one is incorrect. So missense, the question asks for correct statement, right? Okay, missense mutation changes a codon into a stop codon. So this one is incorrect. Okay, it changes if a missense mutation occurs, it changes it code for a different amino acid. The change of codon will code for a different amino acid. So statement yang untuk yang silent ni sepatutnya pergi kepada missense mutation. Alright. So D. Okay, if you look at D, nonsense mutation, no effects on the amino acid produced by codon. So this one is incorrect. So for nonsense mutation, the codon will change. The codon that code for a certain amino acid changes to a stop codon and causes the process of translation to stop immediately. Okay, so this one is incorrect. It produces a stop codon actually. Okay, so that leaves you with C, which is frame shift mutation. It alters the reading frame of the triplet codes referring to codon. That is frame shift mutation. So frame shift mutation occurs due to deletion or insertion of a base. Okay, so for example, you have the sequence AUG, KG, UU, GAC, for example. Okay, so this is the wild type, the original. Okay, the original mRNA sequence. So if mutation were to occur to the DNA sequence and delete this G base, okay, delete the G base, deletion occurs. So it causes the DNA to read the mRNA to be read as AUG. This G will be shifted to here UUGAC. Okay, so this will change the code being read next. Okay, so let's say here you have another G. Okay, so when frame shift mutation occurs, it leads to the synthesis of totally different polypeptide molecule. Okay, it changes the sequence of amino acids, so the effects is more extreme. Okay, because it leads to the change of the codon to be read. Okay, it read into a different sequence of amino acid along the polypeptide chain later. So as you can see, this is the original sequence, so it should read AUG, GUU, GAC, but due to deletion, it will read differently and code for totally different amino acid sequence along the polypeptide chain. So the answer will be C. Okay, that is frame shift. So frame shift can also be due to insertion. Okay, insertion of base. So let's say for example you insert a base A between U and U here. So it causes the codon to be read differently along the mRNA. Okay, question 37. Nonsense mutation refers to the changes in codon sequence that result in nonsense. Stop codon. Changing of an amino acid codon into a stop codon. So the answer would be D. Okay, nonsense. So it changes the code to a stop codon. Okay, so question 38. Figure 4 shows a type of chromosome mutation. Identify the mutation. So this is the original chromosome, and this region of the chromosome is being deleted to form a shorter chromosome. So obviously the type of mutation, chromosomal mutation, is deletion. 39. What are the types of chromosomal mutation represented by 1 and 2? So this is the wild type, the original chromosome segment A, B, C, D, E, F, G. So the first type of mutation, you will observe that there is A, B, C, D, and then you have another D here. A, B, C, D. So you have a repetition of CD region. Okay, CD region you have it twice. So this is what we call it as duplication. Okay, duplication. And then for the second type, the first one is duplication. The second type where you observe that there's missing of chromosome segment. So this is deletion. Okay, deletion. So the region of C and D is being deleted. So the answer will be duplication and deletion, which is the answer is B. Okay. Question 40. Which of the following does not satisfy the Hardy-Weinberg equation? So basically, the Hardy-Weinberg equations predict an equilibrium which is unchanging allele and genotype frequency from one generation to another generation. And in order for the allele frequency or genotype frequency to remain the same from one generation to the next, there's 5 conditions that has to be followed, which is the first one is the population must be large. Okay, the population of the concerned species must be large population. Second, okay, mating is random. There is no preference towards certain feature or trait of an organism. Okay, tak ada preference, tak ada nak prefer. Okay, tak nak rabbit. Contoh, we are focusing on the population of rabbit. Okay, so the rabbit will have to mate randomly. Okay, mating is random. Okay, mating is random. So maksudnya when you have a population of rabbit, so you have white rabbit, you have brown rabbit, you have black rabbit. Okay, so tak ada preference terhadap okay rabbit putih je yang akan membiak sebab dia akan pilih rabbit putih je. Okay, rabbit hitam ni tak akan membiak, so dia akan pupus. So the dominant B allele will be eliminated from the population. So there will be no random mating. Okay, no random mating. Sorry, there is random mating. Okay, random mating. Tak ada preference. Okay, macam tu. Random mating. So number 3, there is no mutation. No mutation. Okay, tak ada mutation. And then number 4, there's no migration. No migration. And lastly is no natural selection. So these are the 5 conditions that a population has to meet in order for the population to be in Hardy-Weinberg equilibrium. So if you look at question 40, which of the following does not satisfy the Hardy-Weinberg equation, which is small population. Okay, small population where the population has to be big. Okay, no mutation, randomly mate. Okay, isolation means there's no migration. Okay, so C is wrong. Okay, that it does not satisfy the Hardy-Weinberg equilibrium. Number 41, the Hardy-Weinberg principle is correct if, okay, the answer would be random mating occurs. Okay, random mating. So mutation occurs, salah, should be no mutation. Genetic drift, we are not concentrating on this one. And this one here, no. Natural selection occurs, should not be natural. There should not be any natural selection. Okay, so the answer will be D. Hardy-Weinberg principle is correct if random mating occurs. Okay, this one here, point number 2. Question 42. In a population, the genotype frequency at one locus are 0.5 for homozygous dominant, 0.4 for heterozygous, and 0.1 for homozygous recessive. The frequency of the A allele in this population is okay. So to do this question, you have to know 2 formula to Hardy-Weinberg equation, which is P plus Q equals to 1. Okay, P is for the frequency of dominant allele. Q is the frequency of the recessive allele. Okay, so 1 is equals to 100%. Okay, because we are concentrating on frequencies. Alright. The second equation that you must know is P squared plus 2PQ plus Q squared equals to 1. P squared is the frequency of homozygous dominant. 2PQ is the frequency of heterozygous. Q squared is the frequency of homozygous recessive, which equals to 1 or 100%. Okay, so these are the two frequencies that you have to know. In the question, it gives you what? Okay, the frequency of homozygous dominant AA, or sama juga dengan P squared equals to 0.5. Okay, the frequency of heterozygous or 2PQ is 0.4. Okay, the frequency of homozygous recessive, which is AA, which is Q squared equals to 0.1. Okay, the question asks you the frequency of the allele A in this population would be, okay. So the question asks you for the value of P actually. P, the frequency of the dominant allele, which is A. Nak tahu kena berapa. Okay, so in this case, the question is really simple, where you will use only this information. Okay, this information. P squared equals to 0.5. So that means the frequency of the dominant allele will be square root of 0.5. You will get what? Okay, so you will get 0.7. Okay, 0.7. So the answer will be D. Okay, so question 43. In a large population of butterfly, 557 are blue while 396 are red. Okay, if the blue is dominant, determine the number of butterfly that are heterozygous. Okay, so the information given here, 557 blue. Okay, blue is the dominant phenotype. So dominant phenotype, and then you have wild 96 is red. Red is the recessive one. Okay, so it means that red is the recessive feature. So it means that you cannot use this information yet, because blue can either be homozygous dominant or heterozygous. Okay, so the value for this is both P squared plus 2PQ. Okay, Q squared is in this value. Okay, Q squared here. So it means that Q squared equals to 369. You want to find out the frequency kan? So kena bahagikan dengan total. Total adalah 557 plus 396. Okay, so the total number of the whole butterfly population would be what? 563 kan, from here. Okay, so 396 divided by 396 divided by 953 to get the frequency of homozygous recessive. So the frequency of homozygous recessive would be what? You just determine, you just press your calculator. So the frequency of homozygous recessive would be 0.41. Okay, 0.415 lah kita buat into 3 decimal point. And then to get the frequency of the recessive allele, you just square root the value 0.415 Okay, so the value of Q would be 0.644. Okay, using the formula P plus Q equals to 1. So you want to find the value of P. So you just insert 1 minus 0.644. So the P value is, so the frequency of the dominant allele will be 0.356. Okay, so the question asks you to determine the number of butterflies that are heterozygous. So heterozygous, it takes the value P to PQ. So now you have the value for P, you also have the value of Q. So 2PQ equals to 2 times value of Q, which is 0.644, times the value of P, 0.356. So you will get, so the frequency of the heterozygous will be 0.458. Okay, so the total number of butterfly population earlier was 953. So you have 2 times the heterozygous frequency with 953. So the number of the heterozygous population would be, okay, so if I press on my calculator, the number will give 436. Okay, the nearest answer here is 439. Okay, so maybe you have to round your answer to 2 decimal point here. Okay, so try for your own calculation, try to round off the value to 2 decimal point, so maybe you get the actual value, which is 439. Okay. Okay, so next question, question 44. In a population of mice, the frequency of the recessive allele for light brown is 0.24. The frequency of recessive allele. Recessive allele is Q. The recessive allele frequency is Q. Q 0.24. Okay, what is the frequency of the dominant allele of black fur? So this one is really easy because you only have to use this formula. P plus Q equals to 1. So you just insert the value of Q. Okay, so P minus 0.24, you will get the frequency of the dominant allele, which is 0.76, which is the answer is B. Okay, so for the next question, in a human population that is in Hardy-Weinberg equilibrium, 1 in 4,000 babies born inherit an autosomal recessive trait. So 1 in 4 babies inherited an autosomal recessive trait. So it means that the value Q squared is already given, which is Q squared equals to 1 over 40,000. So this is the frequency of homozygous recessive. Okay, so what would be the expected frequency of heterozygous? So you have to find the value for 2PQ. Okay, so this is to find the frequency of the heterozygous. So in order to get the frequency of the heterozygous, you need to determine the value of P. Okay, so to get the value of P, you will need to get the value of Q first. So from here, you have to square root 1 over 4,000, square root kan dia. Okay, so the value would be what? So the value would be, the value of Q would be 0.015. Okay, 0.0158 lah. Okay, so the value of P would be 1 minus 0.0158. Okay, so you get the value of P, which is, so the answer is, the value of P is 0.9842. So 2PQ, you just insert the value 2 times 0.0158 times 0.9, untuk masukkan dia yang betul lah, 2 kali 0.9842 kali 0.09 kali 0.9 sorry, 0.0158. So 2pq equals to how many? Okay, so the value that I got from this calculation is 0.0311. So the nearest answer is 0.0313, which is this one here. The answer is B. So maybe you have to round off the number correctly, maybe to 3 decimal point. You can try, okay, to get the exact value of 0.0313. Okay. Okay, so for the last question, so in a population, one out of every 400 people has cancer caused by recessive allele. So it means that B equals to B is the homozygous recessive. Homozygous recessive frequency is Q squared. So this would be 1 daripada 400 orang. 1 out of 400 people has the cancer. Okay, so if the population is in Hardy-Weinberg equilibrium, calculate the proportion of individuals that carry the B allele but are not expected to have the cancer. So you have to calculate again the number of individuals that are carrier. They are heterozygous. Heterozygous is to PQ. Okay, so in this case, you have again to determine the value of P. So you have to get the value of Q first. So Q equals to square root of 1 over 400. So the value would be what? So the value of Q would be 0.05. So you need to determine the value of P using P plus Q equals to 1 formula. So P equals to 1 minus 0.05. So the value of P would be 0.95. Okay, so you just insert all this value into this formula. 2 times, okay, 0.95 times with 0.05. So 2pq, the frequency of heterozygous would be, so the value would be 0.095. Okay, so this is the frequency. Okay, so the frequency for the heterozygous. So the question asks you the number, calculate the proportion. Okay, calculate the proportion. So you have to know how many people that are carrier. So this frequency 0.095 you have to times with the total number of population, which is 400. So the answer will be, okay, so the answer will be 38 people has the cancer. 38 people is the carrier. Okay, 38 out of 400. So the answer will be C. So I hope you know how to apply both of the Hardy-Weinberg equilibrium, which is P squared plus 2pq plus Q squared equals to 1. That is one formula. This is to determine the genotypic frequency. Okay, and then the other one is P plus Q equals to 1. This is to calculate the number of allele frequency. So that's the end of the discussion for part A.